this part. Come on, let's come apart. So you can see inside the cochlea here, this is the cochlear branch of cranial nerve 8. Coming out the rear, coming off the back of the cochlea, cochlear branch, cranial nerve 8. That's the vestibular branch. This is this whole bulge right there. This is the vestibule. Semicircular canals 1, 2, and 3 come off the vestibule. So this is all vestibule. So there's the vestibular branch. The cochlea, there's the cochlear branch. Vestibular branch, cochlear branch. See them merging. They will merge to form cranial nerve 8, vestibular and cochlear nerve. So piece of cochlea, that's the cochlear branch, cranial nerve 8 coming out the rear. So, sectioning the cochlea, we see several spaces, green, blue, pink, green, blue, pink, green, blue, pink, etc. repeated. Each of these spaces is filled with fluid. So we have let's see the term I'm looking for. Well, don't worry about the fluid. So we have fluid-filled space in here. This is the fluid-filled space. This is the fluid-filled space. So green, blue, pink. What does green correspond to here? What does blue correspond to? What does pink? Well, blue, the little space, the space in here. It's called the cochlear duct. Cochlear duct is the smallest of the three spaces. Okay. Then we've got the space here, which is the pink space, all the pink spaces in this model. This is the scala vestibuli. The space down here represents the green spaces on the smaller model. This is the scala tympani. So there's fluid in here, fluid in here, fluid in here. These two have the same fluid. This has a second type, a different type of fluid in it, which you don't really need to worry so much about. So how did I know? So the cochlear duct is the smallest of the three spaces, the smallest of the three spaces. That was easy. But how did I know this is the scala vestibuli? Because this band here is called the vestibular membrane. The vestibular membrane forms the border between the scala vestibuli, fluid-filled space, and the cochlear duct, a different fluid-filled space, which leaves this as the scala tympani. So scala vestibuli, scala tympani, cochlear duct, these two separated by the vestibular membrane here. Let's see. So, we have this structure here, this roof-like structure here. This is called the tectorial membrane. In fact, tectorial actually means roof-like. So tectorial membrane. The white layer here, labeled as number 6 on the model, that's called the basilar membrane. So, what do we find in between the basilar membrane and the tectorial membrane? Notice where we are. The object and the tectorial membrane are in the cochlear duct. Not in here, not in here. So what do we find between the basilar membrane and the tectorial membrane? We find what's called a spiral organ. So actually the tectorial membrane is part of the spiral organ. So we've got the basilar membrane, tectorial membrane, and hair cells. There's an outer hair cell. Actually, I'm sorry, it's an inner hair cell. So it's deep inside, it's under the roof. There's an inner hair cell. These are outer hair cells. So 1, 2, 3, 4 hair cells. One of the hairs, they're actually microvilli that for some reason are called stereocilia. It makes no sense to me why you call microvilli stereocilia. But these are the hairs of this hair cell, hairs of this hair cell, and so on. So the hairs brush the tectorial membrane. It's the hair cells that are involved in this case, in the cochlea, in audition, hearing. There are also hair cells in the vestibule. There are hair cells at the base of each semicircular canal. There are hair cells that are involved in equilibrium. So, the cochlea is involved in audition, the sense of hearing. So the cochlear branch conducts active potentials for the sense of hearing. Let's see. What else do we have here? So what actually happens is, as sound waves strike the tympanic membrane, those sound waves will cause the fluid inside these three spaces to begin to slosh back and forth. To put it simply, causes the hair cells to bend back and forth. So now they're brushing against the bottom pectoral member. If they bend in one direction, gated ion channels close. If they bend in the other direction, gated ion channels open. So you've got mechanically gated ion channels, mechanically gated potassium ion channels. And they bend in one direction, they open up, potassium flows in. Remember from earlier this semester, potassium ions are in greater concentration inside cells, lower concentration outside cells. Not here. It's the weird exception. Why? Because